And again, welcome back to the virtual March of Dimes High School Science Week. My name is Monica Wirt-Parkinson. I'm the Director of Public Programs for the Salk Institute, and I'm really happy to be with you this afternoon. High School Science Day is a favorite Salk tradition. In fact, this year is our 31st annual event. And while we're very sad we can't welcome you onto campus, we're still so proud to be able to offer this event in a virtual forum. All of our Salk researchers are really looking forward to this event, and they're so excited to interact with you. So really briefly, the Salk Institute is not an acronym. It's actually named for Dr. Jonas Salk. You may have heard his name in the news recently. He was actually a key player in another public health crisis, the polio epidemic, which affected children in the 1940s and 50s. Dr. Salk developed the first safe and effective polio vaccine. He then went on to pursue another dream, which is to create a collaborative environment where researchers could work together to explore the very basic principles of life. 60 years later, the Salk Institute is a world-renowned basic biological research facility. When designing the Salk Institute, Jonas Salk selected architect Lou Kahn, and he famously told Lou Kahn to build him a facility worthy of a visit by Pablo Picasso. Many years later, artist Françoise Gillot was in San Diego visiting with some friends, and they invited her on a tour of the Salk Institute with Jonas Salk. Needless to say, Françoise Gillot and Jonas Salk were quickly married 6 months later, and they stayed married until his death in 1995. Jonas Salk quickly saw that the original buildings, which you see in the distance, were being outpaced and there was a need for additional space. So in the 90s, he worked to develop and commission these east buildings that you see here. And actually, Françoise Gillot, a very famous artist in her own right, commissioned paintings which she sold to help raise funds for the building. And we're very excited now to have these new buildings because they house meeting space, as well as additional space for administrative and labs, and a lot of our core facilities. So today's format, just to go over very quickly, we're in the introduction right now, and then we're going to move on to the virtual lab tours. Each lab tour is very unique and created by the scientists themselves. So we definitely encourage you to attend more than one lab tour. You may find a topic of interest that could spark some additional interest for you down the road. And each tour is designed to be standalone. Each of the links are going to be separate for the lab tours, and we'll be sending those out to you not too long after the event concludes, probably sometime next week or the week after. So you'll have a chance to go back and watch any lab tours that you may want to see. We really hope that everything runs smoothly, but in the event of any technical issues, please have some patience with us and send us an email to education@salk.edu with any additional questions you might have. So this panel discussion is really driven by you. We want to hear what you have to say, and we want to hear your questions. So it's very simple to submit a question. Just go ahead and look for the link that's right underneath the YouTube video, and you can click on that where you can submit a question to an individual panelist, or you can submit a question to the lab overall. If you want to learn a little bit more about what the lab does or what the panelists do, you can click on the Show More button as well to learn a little bit more about the panelists. Before we launch into today's lab tour, I wanted to tell you a little bit about some of the high school programs that we have going on above and beyond the March Dimes High School Science Week. We offer an 8-week paid internship under the High Top Brody High School Summer Scholars Program. You attend a biotech boot camp and then spend 7 weeks in the lab with a Salk scientist. It's a great opportunity to conduct real-life research and develop science communication skills. We're very hopeful that the program will happen this summer of 2021, but it's contingent upon public health guidelines. So keep checking our website for regular updates. We should have a program status update in hopefully early March. A program that we will be happening no matter what is our Introduction to Research Science and Communication virtual program. It's a 5-week professional development opportunity, and because it's virtual, it's open to students all over the country, not just San Diego students. It's about 5 to 10 hours a week of mostly asynchronous work, and it takes program elements from the High Top Brody High School Summers program, including reading journal articles, analyzing data, preparing scientific presentations, and then preparing a final capstone project. This program will be taking place in the summer of 2021, and our program lottery opt-in is actually open right now. It is open through March 31st, so if you're interested in applying, we definitely encourage you to check that out and fill out the registration form. If you have any questions about the program, you can email us at education@salk.edu or visit salk.edu/education, and we'd be happy to go ahead and answer any of your questions, and you can learn a little bit more about the programs. And as a quick thank you to our donors, the March of Dimes High School Science Day is really made possible through the generous support of Ann and Neil Blue High School Science Fund. And our sincere thanks to the Blue family, all of the supporters, partners, and volunteers that make education outreach possible. And really thank you to the March of Dimes. So today we're really excited because we'll be touring the Stem Cell Core facility. The Stem Cell Core facility is a little bit unique because it's a specialized lab on the Salk campus that has cutting edge technology, and they work with scientists all across the campus to help plan, oversee, and run their experiments. And we hope that you really enjoy the chance to take a sneak peek into the video. It may give you some ideas of what a life in a scientist looks like and some of the options that you can see and career options when you're working in the science field. So with that, I welcome you to enjoy the Stem Cell Core video.
Hi, my name's Ken. Welcome to the Salk Stem Cell Core. Here at the Stem Cell Core, we focus on providing stem cells as a resource to researchers here so they can utilize them in studying human disease. Here at the Core, we do this in three main areas. First, we provide the actual facility to work in. If you look around here, you can kind of see the space we're in. Here's an area we've devoted to preparing a specialized media for working with stem cells, and this is our technician, James, and he's helping us make this media today. Another key thing we provide are reagents. Stem cells are incredibly finicky, so it's important to have the right materials to work with them. And so we spend a lot of our time focusing on finding the best reagents and producing those in-house to make those available to researchers here. The last major resource we provide is training. As I mentioned earlier, stem cells are difficult to work with. And so if we can train researchers on best practices on how to utilize these cells, it improves their efficiency here in the lab. Let's hop into a tissue culture bay and check out some cells. This is one of our tissue culture bays here at the Salk. Let me briefly introduce you to the space. There's kind of three key pieces of equipment we use in a tissue culture facility. One of the biggest components are these laminar flow hoods. These are essentially sterile cabinets that we can work with the cells in without risk of getting those cells contaminated. Another key thing are these CO2 incubators. These incubators are essentially a cell hotel. The cells live in here. We humidify the space, keep it at 37 degrees Celsius, which is body temperature, and also inject a small amount of carbon dioxide into the chamber, which helps out with gas exchange and controlling pH. So the cells spend their entire day in here. The last major thing we need in a cell culture room is a really simple, basic microscope. We have to look at these cells daily to make sure that they are not contaminated, that they're growing properly, and that they have the right visual characteristics, something we call morphology. So we do this every day, and we utilize these three pieces of equipment in this room to work with these cells. Let's check out some cells. The first cell type we're going to look at are embryonic stem cells. Human embryonic stem cells are pluripotent, which means they can generate any of the cells in the human body. In the context of research, this is hugely valuable to us because we can take these cells and turn them into heart or skin or neurons, and we can start modeling human disease with these cells in a dish. Let's check out the cells. So what we're looking at here are embryonic stem cells. These cells grow in a structure that's called a colony. And you can see the structures in the middle of the plate here, these large round structures. That's not a single cell. That's actually thousands of cells smashed together into a single unit that we call a colony. Pluripotent colonies have three distinct characteristics. First, the boundary of the colony should be extremely defined and very, very uniform compared to the background of the plate. The second major characteristic is that the cells inside the colony are extremely small. Here we're looking at 5x magnification, and at this magnification we can barely see the boundaries in between the cells. So very, very small and compacted inside that colony. The last major characteristic for pluripotent colonies are that they have a very, very large nuclei compared to the rest of the cell body. We call this a high nuclear to cytoplasmic ratio. In the context of pluripotent stem cells, this has a functional purpose. Since these cells are primed to turn into any of the cells in our body, they have an extremely wide open genome to facilitate the machinery in the nucleus that turns on and off genes. As cells differentiate, the genome actually collapses down in a more restricted space to restrict the access and restrict the number of genes that can be turned on or off. The next cell type we're going to look at are some neurons. And the neurons we're going to look at actually came from embryonic stem cells. They started as embryonic stem cells, and we turned them into neurons through a process called differentiation. So the cell types we're looking at now are neurons. And as I said, these were derived from embryonic stem cells through a process called differentiation, where we essentially force the embryonic stem cells down a developmental lineage to turn into a mature neuron. Neurons look distinctly different from the embryonic stem cells we were looking at before. We've completely lost that colony morphology, and now we have individual cells that are forming these networks. Neurons operate by passing around electrical impulses in the context of our central and peripheral nervous system. Here in the dish, they're actually doing this now. If we put in a special dye, we could actually visualize electricity passing between these cells in this dish. Neurons do this trick of passing around electricity by having two key parts. One part is a listening in, where an electrical impulse is received, and the other part is a speaking in, where that electrical impulse is passed on to the next cell. The last cell type I want to share with you today are cardiomyocytes. These are a really unique cell type. Again, these were differentiated from embryonic stem cells. At this time, instead of directing these cells into neurons, we're treating the cells with a different combination of chemicals and turning them into the heart muscles of the cell. Cardio means heart, myocyte means muscle cells, so we're literally looking at heart muscle cells. These cells have a really, really unique morphology in that they physically pulse in the dish when they mature. And so if you look here, you'll see that there's this rhythmic beating that these cells are doing. As these cells differentiate, they're actually self-organizing themselves into muscle fibers, and then they can pass along an electrical impulse and do this contraction. And if you look here, you can see that it's somewhat organized. One area of the plate will beat, and then following that, other areas will beat, similar to how the chambers in our heart would be organized. So now that I introduced you to those cell types, the pluripotent stem cells, the neurons we differentiated from them, and also the cardiomyocytes, let me quickly show you our cryostorage system. Cryostorage allows us to physically freeze cells. Oftentimes when we generate these cell models, it's really important to be able to bank these cells down so that we can utilize them in the future. So let's check it out. So what you see here are our cryostorage tanks. These are very, very well insulated chambers that we can pump a very small amount of liquid nitrogen into the bottom of. That liquid nitrogen allows us to cool these chambers to a very low temperature, roughly negative 190 degrees Celsius. That's almost 200 degrees less than the freezing point. So let me pop open one of these containers and show you how this works. So in each of these containers, we have 12 racks. Each of those racks holds roughly 12 boxes of cells. Each box of cells is a 9x9 box holding roughly 89 vials. So a really large capacity for storage of material between these two freezers. Here's an example of one of those cryostorage racks. You can see all the boxes aligned in there. Each one of these boxes is specific to a project that we're working on here and has a unique cell type in it. We'll quickly open one up and show you. So you can see all the vials in this box. Each one of these vials is filled with cells in a cryogenic state. When we want to utilize these cells in the future for research purposes, we can take these vials and thaw them and put those cells back in culture, and they work similarly to the cells you saw earlier. So here at the core, one of our missions is to provide a large tissue culture environment for people to do research in. We currently have roughly 40 different researchers here from the Institute from 12 different labs. So a lot of different labs and a lot of different people utilizing this space. So we have to make sure we have a lot of resources for them to use. So let's quickly check out some of our other tissue culture spaces. This is a few of our researchers. This is a member of our Gage lab, Abed, and his intern, Sarah. They work on neural disease. So this is another one of our tissue culture space. If you take a quick look around, you can just see that we're set up the exact same way we were in the other room. We still have the incubators, microscopes, and hoods. And you can see a few of our users working right now. This is another example of one of our tissue culture suites. Again, having a lot of different hoods and incubators for our researchers to use to do this high volume of work. Well, thanks for visiting the core. I hope you learned something about stem cells and how we can utilize them for research purposes. Have a great one. Hi, everyone. Thank you so much for joining us today. We're all really excited to be here with you and answer some of your questions after the virtual tour here. I'll quickly introduce myself, and then we'll have the rest of the panel introduce themselves as well. So you have met me virtually already. I'm Ken Diffenderfer. I'm the director of the Stem Cell Core here at the Salk Institute. I've been working at the Salk Institute for almost 10 years now, and I've been in the Stem Cell Core for all those 10 years and working directly with human stem cell populations and other human cells for the purposes of generating unique and relevant disease models for our researchers here at the Institute. We'll go into that in a little bit more detail. I am originally from Georgia, but transplanted to Southern California when I was in middle school, and have been in and around Southern California for all of my kind of academic K through 12 life, college, and then also professional life since then. As well. My role here at the Institute is really overseeing the core facility, which is a unique hub of activity where we can kind of develop these unique stem cell models and be a source of expertise to disseminate those out to a larger audience here at the Institute, and also to kind of constituents that we work with locally and or internationally, nationally or internationally. So kind of a unique opportunity for us to do some really exciting stuff here. So to continue with introductions, I think we'll have Amy go next.
Hi, I'm Amy Pekinen, and I work in the Stem Cell Core. I've been at Salk for almost 7 years now, about 6 of them in the Stem Cell Core. I used to work in the Gage Lab, which is where the other 2 panelists are from. I grew up in Temecula, California, and I've pretty much stayed in Southern California for my education. And so what I do in the core is I do a lot of the day-to-day upkeep, ordering and making sure it's running well. And then I also do projects where I will do stem cell work for researchers if they either don't have time or the resources to do it on their own. So they'll hand them off to me and I'll take care of them.
Awesome. Thanks so much, Amy. And next we'll have Jeff introduce himself.
Hey everyone, I'm Jeff Jones. I was originally born in Florida, and then I moved up to Wisconsin to complete my PhD. So that's why my title says postdoctoral fellow. And that means that I have the expertise to run projects, design experiments, and train up-and-coming scientists within a bigger lab. So I'm in the Gage Lab along with Thomas. When the goal is to kind of get my own lab at some point and design my bigger picture science projects.
Thanks. Awesome, Jeff. And then Thomas, last but not least.
Hi, everybody. My name's Thomas. I've also been at the Salk Institute for about 6.5 years now. I'm originally from Columbus, Ohio, so I came out to UC San Diego for my undergraduate work, fell in love with working at the Salk, and they haven't managed to get rid of me yet. My role in the Stem Cell Core is that I rely on both Ken, Amy, and the various staff there to help me design, support, troubleshoot, which happens a lot, the ongoing projects that I have. So I'm interested in understanding neurodevelopmental disorders like autism spectrum disorder. And we do that by actually growing a miniature version of the brain, or as close as we can get right now. So what Ken said about these cells being very needy and difficult, it's an incredible resource to have them on staff, available. I don't want to say 24/7 because they do have work-life balance, but to help us out, guide us, and train us on these advanced techniques so we can answer our own scientific questions.
Thomas, can I put you on the spot real quick? I think you left out what's next for you in life. That's an exciting note. I think we should.
Absolutely, yeah. I'm flexing it in my title down there. So I'm so interested in this research and specifically how it applies to people and patients across the world that I will be pursuing an MD PhD program where you become a doctor and then you also become a scientist, and you kind of mishmash them together to try to discover something that can make medicine a little more effective or develop new therapeutic strategies for things that don't quite have a treatment that works yet.
Yeah, awesome. Thanks. Okay, cool. So let's get started with some questions. And I think right off the bat, to kind of center the conversation a little bit, maybe have a little bit of a roundtable around the concept of disease modeling and maybe what that means to you and how you specifically engage in that. And maybe we can take the 3,000-foot view of that topic for the engagement portion. And maybe, Jeff, if you don't mind, I'll put you on the spot to kick us off there. So what does disease modeling, mean to you, and how are you engaging in that directly?
Yeah, absolutely. So disease modeling is pretty much as it sounds, trying to take a complex disorder, and in my case, I'm interested in Alzheimer's disease, and be able to replicate some aspect of that disease in a petri dish so that we can use that to study. And in the context of this project, for this panel, we can take patient-derived pluripotent stem cells or embryonic stem cells from an embryo, and you can go to patients, gather their skin cells, and reprogram them to a pluripotent state. And these are called induced pluripotent stem cells. They're very similar to embryonic stem cells. And that's where the STEM core staff really comes in. They coordinate with clinicians to get these cells, they do all the reprogramming, and then I can use the cells that they generate to model Alzheimer's disease in a dish, generating the various cell types of the brain to see if there are cell-specific deficits, patient-specific deficits, et cetera.
Awesome. Thanks, Jeff. And to quickly clarify for you guys in the audience who might not be as familiar, induced pluripotent stem cells is a relatively recent technique and technology that allows you to take a permanently developed, permanently mature skin or a cell from a patient through a biopsy, through a blood draw, and then we can use technologies to essentially reprogram that cell to very, very early stages in human development. That would be the pluripotent state, where it could then become any cell type in the human body. And so we can effectively turn back developmental time on cells that are in your body right now, and then use that as a jumping off point to create really unique and powerful modeling systems. And all of this hinges on this idea that the genetic code that you have, the A, T, C's, and G's in your genome, confer unique things about you, right? And in some cases, that is a disease or disorder. And we can capture those A's, T's, C's, and G's in your blood cells or skin cells, turn those into these induced pluripotent stem cells, and then model a disease or condition because the same genetic characteristics are captured in those cells. This whole idea of doing this, we refer to often as connecting a genotype, which is something that is encoded in your genetic information. And we use that genotype to study a phenotype. And so connecting genotype to phenotype. And a phenotype is really anything we can observe and measure, right? We can think of this as a large scale thing like your weight, your height, your eye color. Or we can think of it as something incredibly small, like, how much insulin is your pancreas secreting at a given time on Thursday afternoon, right? And we can even take it down even smaller to there. So it's just something we can measure. And so the real utility of this technology is connecting these genotypes, A's, T's, C's, and G's in your genome, to a phenotype, something that we can measure that would be relevant to a disease, right? So just wanted to take a step back there and emphasize that. Thomas, how about you next? What does disease modeling mean to you, and how are you specifically engaging in that?
Absolutely. So for us, we're very similar to what Jeff is working on, but we're coming at it from a different angle, which is early on in development. So autism spectrum disorder is something that is still really complex, convoluted, and very dynamic between individuals. So for us, disease modeling is all about seeing actually how the disorder grows and progresses over time. We're not really interested in what it looks like when it has fully developed. We're trying to figure out how it occurs, when it occurs, and what factors influence it. So to do that, we actually have a miniature system that kind of faithfully recreates development in the actual human brain as you're gestating up until birth. And we can see how neurons move, migrate, grow, expand, divide, and all these wonderful functions that are really tightly regulated. So we want to start seeing maybe these neurons divide a little too early. Maybe they migrate a little too far. Maybe they're forming connections in different parts of the brain we wouldn't normally find them. So we're all about that pathway from the beginning and the onset of these neurons being born, being generated, and then what they end up doing and where they end up going.
Awesome. And then Thomas, since we're on it, I think you kind of briefly touched on maybe a topic that has a broader implication and impact here, and that's organoid technologies, and that's obviously something you've been involved in. Can you briefly take a few minutes just to explain organoid technologies at a basic level?
Absolutely, absolutely. So we had a wonderful tissue culture invention a few years ago, which was generating cells that we're interested in from these iPSCs, right? So the idea was that if you're interested in neurons, you can make neurons from a patient stem cell line. And we made them as a two-dimensional layer, kind of like a piece of paper. And the cool thing about that is that we actually had neurons. But if you think about your brain, you've ever seen a picture of your brain, you know it's all squishy, folded up, it's three-dimensional, right? And the neurons in your brain are actually forming three-dimensional connections. They're projecting front and back, left to right, up and down, they're all over. So when you grow something flat, you don't really get the full potential of the connections they could be making and the full level of complexity that would actually exist in a person. So organoids are currently the closest we can get, and they're actually these three-dimensional balls of cells that really, really coolly can self-organize with only a few different factors provided by us. They'll start forming some folds, they'll start forming buds, and they actually get close to forming the morphology of the actual brain. So it's much closer to what the organization would look like in you, for example. And that is a very exciting opportunity for us to learn a lot more new aspects of neurodevelopment.
Awesome. Yeah, it's an incredibly new and exciting realm that is a very hot topic in the stem cell community. And it all centers around this idea that we are complicated and complex, organisms made up of organs that are organized into 3D space. And so why can't we transition our cell models that we traditionally use to model human disease and development from, something we do more traditionally in a 2D context, like Thomas said, cells growing on a flat surface, to cells growing in a three-dimensional space and allowing them to organize and develop in that space to, again, capture those more complicated relationships that they would have and model a more complicated system. Awesome. Thanks, Thomas. And then Amy, what does disease modeling mean to you, and how are you engaging in that on a daily basis?
Well, uniquely in the core, we're not asking specific questions, so we don't actually look at a specific disease to model, but we are helping all of our researchers that utilize the core in their disease models. So just depends on which project that I'm working on at the time, on which disease model that we're trying to study or replicate. But my major contribution to the research is I'll be the one that makes the stem cells for the researchers. So I'll reprogram them. We'll have it organized where the clinicians deliver us a sample of either skin or blood or whatever somatic, fully differentiated cell that they can get from the patient, and I will reprogram them into stem cells for the researchers to utilize in their research.
Awesome. Yeah, awesome. Thank you. And just to kind of put that in a little bit more context, Amy and I work directly in the core, and we're generating tools for researchers to use. And those tools are used by people like Jeff and Thomas, who are two researchers that we support. And so Amy and I are generating tools for our researchers to utilize to answer very, very specific questions. So Amy and I might not have very specific questions we're interested in answering as far as human disease development or aging, but Jeff and Thomas do, and we can make sure they have all the resources at hand to do that as efficiently and effectively as possible. So we do have another question that popped up here, and it's in regards to time. And I think there's two different ways we can look at this. So and maybe this is a little bit more relevant for Jeff and Thomas, as you guys look to kind of do a new experiment, how much time are you spending planning it? And then the follow-up to that is, how much time does it actually take to do it? Maybe we'll start with Jeff.
Really depends on the experiment. I will say generally that time is a huge factor, and the strength of having a core facility like the STEM core is that it takes the time away from generating all these lines, managing all the cell lines that they've made, and making sure everything's cataloged and easily accessible. So the fact that they help with that gives me more time to design experiments. To directly answer the question, there are some experiments we can do with just a few minutes of planning, but then other experiments require literally months. I mean, we are researchers at the end of the day, so you can come up with a really interesting scientific question, and then you have to see, okay, has this been done? I have to read up on this particular aspect of this question, and then you have to come up with technical ways to address that question. And if it's something you haven't done before, that can take a long time to learn what's the best way to test it. Is this the right readout? How do I know that the results are representing reality and not some kind of artifact? So anywhere from a few minutes to a month plus.
Yeah, awesome. One of the things I really love that Jeff kind of alluded to and touched on is that oftentimes we kind of view the research process as being really linear. And so we have a very specific question, we ask that, we answer that. And the reality is that is never the case. There are a lot of roadblocks. There's a lot of failing that happens in that process, and we're constantly redesigning, reapproaching, and reinitiating experimental models. And a successful research program is really happening under a lot of failure, right? And then after you've had things that don't work, you ask critical questions on why and how they didn't work, and then you can go from there, and that builds and builds and builds until finally you have something that affects people's lives, right, in a more direct way that we can kind of share with the broader research community. Thomas, how about you? What do you have any other insights to add there, just on kind of general amount of time that it would take to design or implement an experiment?
Jeff and I are living in very different worlds. On the student end of things, we have a lot of moments where you're in lab, it's late at night, you're like, what if we did this? And especially in neuroscience, there's so many unanswered questions that you could design an experiment just like that. You think, this is a wild idea. No one's done it. Let's just try it. We'll see what happens. That is a little bit of science without direction, where you think something sounds exciting, you think something sounds promising, and you just go for it. And on the other side, there are experiments, just like Jeff said, that we plan for almost up to a year. And in the context of organoids, it can take me 3 months to generate one batch of samples. So over those 3 months, you kind of always got to be rolling with the punches. Potentially, they don't work. And you have to have another experiment going on the back burner. Or you get those samples, and that's your one batch. You better have a bunch of experiments ready. So you're kind of always oscillating between doing the work and planning the next step, doing the work, planning the next step. And you want to make sure you have not necessarily a linear idea of where you're going, but that you have an idea of what comes next so that you can be prepared to move on to the next experiment after you finally get a batch of stem cells that works.
Yeah, I really love that. You hit on that, Thomas, and that anticipation that we all have to be constantly engaged in. If you ever get to the point where you're not sure what to do, you have missed a moment and are wasting precious time and resources. Yeah, that is absolutely critical and a constant challenge, always to be a few steps ahead.
Well, I think Thomas
in a very quickly moving field. Sorry, Jeff, what was that?
Yeah. I was just going to say, I think Thomas brings up a really important point in that no matter how much planning you do, at the end of the day, we're asking biological questions, and biology is very complicated. So you can come up with a perfect experiment, you can test it out, and then it doesn't work. And then you spend the next month or two figuring out why didn't it work? Is it a technical problem, or is it actually reflective of the biology? And so that's just the life of a scientist.
Yeah.
Was it me? Did I mess up?
And the answer to that one, Thomas, is yes, it was. In that specific context. Okay, cool. So we have another question here, and I love thinking about this. So, I mean, all of you have a good amount of experience and have been doing what you're doing for a very long time. But if you had to, reach back in time to your more naive self, what is kind of one thing you wish you knew then that is just absolutely routine for you now? Maybe we'll start with you, Amy.
Well, one thing that I wish I would have gotten better at in the beginning was I wish I had a better planner. Like I remember putting things in a notebook just all willy-nilly, and like you go back a year or two later, like, when did we do that experiment? What did we do? What cell line was that? And you're like searching through your notebook like crazy. I wish I had a better system in the beginning. It would have made life a lot easier in the long run.
Yeah.
Being organized more.
I think we would all agree with that sentiment. There's not, as research scientists, the ultimate goal for everything that we do is that it's reproducible. And what that means is essentially, we or colleagues or people on the other side of the world can repeat what we did, right? And show that it is still impactful and relevant, right? And it's through that reproducibility that research science really, has its impact and has its influence on a larger community. And if you aren't taking good notes and you don't have good records, there's no way to reproduce things, or there's no way to repeat it, right? And so I think it's something that we could all say we don't do very well, and we could always do better. The lifeblood of what we do is driving that reproducibility. Thomas, Jeff, do you guys have anything to add on the context of reproducibility?
I would definitely echo both of your sentiments. Organization, time management, note-taking, all of those things are really critical that you kind of overlook starting out. The only other thing I would add is maybe just perseverance is a really good quality. Don't be too discouraged, because at the end of the day, failure is most of our job. Most hypotheses don't work out, and so that's okay. And it can be frustrating, but eventually the hypothesis won't work out, and it tells you something really interesting that you weren't expecting, and that can be very exciting.
Yeah. One thing I always like to think about is that, when things don't work out, it's only a failure if I don't ask the question of why it didn't work, right? We're still learning something. We just have to ask the next question to learn that, right?
Right.
Yeah. Thomas, how about you? Something that you know now that you really wish you knew then?
Absolutely. I got a twofold, a little more philosophical. First, no one ever told me that you have multiple rotating hypotheses. We talked about earlier, there's failure, there's compensation, you got to roll with the punches. No one really ever explained to me that your first hypothesis is a guess. It's informed by literature, it's informed by what we know, but it's never been done. That's why it's your hypothesis. So you don't know what's going to happen. And the way you actually do science is by asking a series of questions, a series of smaller questions that lead you to one big understanding or statement that kind of connects all of them together. So when you're writing science, when you're communicating science, you share a story. You're not sharing a finding. You're sharing a series of findings that explain something in a new and unique way. And that kind of folds into my second point, which is that nobody ever actually told me that just because you may know less about a field, or you may be newer, this is particularly applicable to me as a student, you may know less than your PI or someone who's been conducting this research for 20 years, that doesn't make your ideas less valid. And in fact, the insight and the input of someone who sometimes has less expectation or less background knowledge can be just as valuable, because there are things the field looks as like, nah, that could never be it, never be it. And it takes someone to come around and say, I don't know, maybe it is. So if you feel that you have a weird idea or you have something to contribute, no matter your level of education or experience, it's always great to ask questions and to talk about it and say, I think this could actually be why. Because that kind of insight is not really restricted to age, experience, or education, in my opinion.
That's a great point. And by the way, the textbooks that we read are basically rolling hypotheses. It's the best guess of how it works.
Yeah, absolutely. Yeah, it's what we know now, right? Mm-hmm. And things change very quickly, and textbooks don't, right? Yeah. That's a great one. One thing I wanted to add too, just on this topic, is the one thing I really wish I knew when I was younger and getting started in my academic career, with undergrad, with college, high school, and even early on in my professional career, is I had a major failure in accessing expertise that was around me, and so I was always really bashful about talking to mentors about things, reaching out to faculty. They just seem so important and so beyond me, and they obviously want nothing to do with me, and I'm wasting their time if I write them an email. And the reality is that's why these people are here, right? If you have committed a life to being a professional in the academic research scientist, you're one of your primary jobs, one of your primary goals is to support that next generation and provide that training and support to them. And I kind of wish I was more active in reaching out to those connections early on in my career and seeking out those opportunities, building that kind of network that ultimately I have now, but could have gotten a lot sooner and maybe possibly a little bit more organically than I had done now. So I will add that as well. So we have another question here, which I think is kind of a fun one to think about too, and it's revolving around this idea of how do we communicate our science, either in the written form or other forms. And so maybe the specific question could be is kind of what is a day in the life of you communicating your science either in the written or other forms. And Amy, I think we'll start with you again. And maybe we can think about like maybe some of the minutia of, standard operating procedures and protocols that we develop there.
Oh, yeah. One of the big things about the core is that we'll develop, validate, and test protocols for various products, various procedures dealing with stem cells and how to, keep them alive, basically. And a big part of the core is training. And so we'll get new scientists, we'll get undergrads, we'll get, veteran scientists that are just new into stem cells. And we have to communicate how to work with a very finicky cell line that not a lot of people have experience with. And so we have written protocols, we have, I'll walk them through it, we have training. So that's how we communicate through that a lot. And then troubleshooting. Every day I'll get someone that walks into the office and, hey, Amy, can you look at these cells? Is this normal? And so, yeah, it's a lot of it's Thomas.
Yeah, a lot of that troubleshooting is Thomas, isn't it?
Yeah. And it's always, no, it's not normal.
Yeah. So I guess to give that in a more like 3,000 foot view in a nutshell kind of context, a lot of the communication you do is very technical, right? It's what are the specific, step 1 through 55 that have to be done to make this work, right? Yeah. And, if Thomas has an issue that came up at step 23, what are some, side steps that, we would have to recommend in there? So a lot of very technical stuff that we communicate on. And a lot of this is written down into formal protocols, which we disseminate to our user base and have available to them. And some of it happens more informally just through in-person interaction, email, etc., just to get kind of some of that expertise disseminating out. Hey, Jeff, maybe you, how do you find yourself communicating? I know you are, a little bit more involved in the grant writing side. So what do you feel like you do most for your scientific communication?
Right now, it's definitely grant writing. So I'm kind of taking that idea of rolling hypotheses that Thomas alluded to, where maybe we've done a couple of experiments. They didn't turn out the way we expected, but we got some interesting results and we don't really know what to do with that. Or maybe it gives us an immediate idea for some cool experiments to do. So then I take the data and I have to formulate a new hypothesis and try to get money to actually do the experiments to test the question. And so I have to communicate, one, the idea itself, and then why it's important to do. And hopefully someone agrees and they help us to fund the projects.
Yeah. Jeff, out of all the grants you've written, how many have actually been funded?
I actually have a pretty good average right now, over 50%.
Okay. Wow.
Most people, I would say the historical record, you're doing well if you're in double digits at all.
We need to keep Jeff around, guys. He apparently has a inroad here at NIH for some grant funding. So, Thomas, I know you maybe just on the student side mostly, but how do you find yourself mostly communicating science?
Yeah, I'll throw 2 different perspectives out there. So first is I teach biology courses at UC San Diego. So a lot of my communication is with undergraduates who might not have a ton of exposure to what we're working on. And someone who's not going to have the 6-year experience of saying, oh, have you ever heard of a GPCR and the different kind of hypotheses where people just throw out random letters? They're like, I don't even know what language you're speaking. So a lot of the communication I do is simplifying these concepts down and saying we're interested in how the brain grows. We're interested in how these neurons pathfind and basically drive around the different regions of the brain to find their targets. And my communication focuses on making that essentially more accessible to people who are not directly doing the work or who are not directly involved in the project, which actually has a very, very nice side benefit. That means when I run into Amy, when I run into Ken, Jeff doesn't count because Jeff is in my lab and knows pretty much what's going on. But any of these other resources that Ken was talking about, these other researchers, you run into a wall and you can say, none of this worked. What do you think I could do? And you could have someone who's working in a completely different system who's working in mice. As one of my best friends in the lab works with mice mostly, and it's just like, I don't know, maybe this is what we do. Why don't you try this? And I communicate essentially the basics of what I'm trying to observe, what I'm trying to quantify, and I can get advice from people who don't even work with what I work on.
Yeah.
So a lot of that communication is with my peers and troubleshooting too.
Yeah, awesome. I love how you can kind of interconnect those two ideas, Thomas, that, that more lay communication that you would do with students on the education side really informs you and how you can more effectively communicate something on a much more technical level, right?
Absolutely.
I really love that connection. We often kind of separate those, and they're very much interconnected. At the end of the day, it's all about telling a story, right? And communicating a bigger idea, a bigger concept in a clear and concise way. And whether we use that with jargon-laden technicality or in a more, user-friendly kind of accessible format, the goal is always similar. It's always the same. Awesome. So we do have another question here. I think, Thomas, we get to pick on you some more. Would you mind going into just a teensy bit more detail on the organoid process and kind of how you start with a induced pluripotent stem cell, and then how do you end up with this amalgamation of different cell types that somehow replicate the brain in 3D space?
Absolutely. So if it weren't so interesting, it would definitely be a nightmare, because it basically takes the whole process and just adds another level of complexity. The beauty of it is that it's surprisingly simple. So we take the stem cells, we grow them flat, just like everybody else does. And then we use a special enzyme that can just kind of scrape them off the plastic they're living on. And when they get scraped off, they're just floating around in the media. And just by how they float, they start to pull together into these little spheres. It's just kind of the natural form that they go for. They actually stick to each other. They're a little sticky. So you end up with these small little spheres that are floating around. They're still stem cells, but now instead of being flat, they're like little beads. And we can start to add chemicals and messengers that say, hey, be a neuron. Or we can get even more specific and say, now become the front of the brain kind of neuron, because we have different regional identities in the brain. So we go through these steps one by one, saying, okay, first we're going to make you basically any kind of neuronal something. And then after a few weeks of that, they're swimming around in the media, they get a little bit bigger. We say, okay, we're going to drill deeper. Now that you're a neuron, let's make you a specific kind of neuron. So we change it up, we add a new media, new chemicals, new factors, and we get closer and closer to the specific region we're interested in. And then we can actually combine these different little floating spheres. Let's say we make one that's similar to the back of your brain. We can make one that's similar to the front. We can stick them together, and because they're sticky, they'll start communicating, and they'll actually grow into each other and form a network that's very similar to what's happening in your brain, where they're actually shooting axons in different directions saying, hey, I'm the back of the brain, hey, I'm the front of the brain. And if you poke them and stimulate them, they actually respond, and you can see the circuitry evolve. So the most complicated part of this process is just going from the flat stage, lifting up, and forming these balls in the proper way, because it is, for some reason, very finicky. If the colonies or the two-dimensional cells are not happy, we tend to give them emotions. They look sad, they look upset, or they're happy. Then you get little pancakes, you don't get the spheres, and there's all this stuff, all this troubleshooting. But the process, when it does go correctly, and you have good cells, and you have good reagents, is actually fairly simple. It's just creating these three-dimensional clumps.
Yeah, absolutely. One thing I love the most about that is this idea that cells know what to do. And our job, in many cases, is to get out of the way, right? And just put them in a situation where they can do what they do, right? They have the program. They know how to make a neuron. We just have to let them do it.
Absolutely.
It's a really kind of liberating idea that they are the experts and we are the naive arbitrator of that activity. Awesome. So I think we have a really good question up next here. And it is what initially got you interested in this career path. I think, Amy, let's start with you. You have a muddled career history that I think...
I have a very twisty-turny career path. I didn't know that I wanted to go into science when I was in high school or, when I began college either. I actually started my degree in psychology, and then I realized really quickly that that wasn't for me. And I had an entire career in culinary. I was a sushi chef for like 8 years, and that wasn't for me anymore. And I decided to go back to school, and I reassessed on what was interesting about life, what could I contribute to society, how best to have a fulfilling career, and it landed on biology and science and learning more about how we work. And so I found programs in college that helped me get closer to a career path that I would have been happy, that I'm happy with. And that's where I went. And I landed with stem cells. I got into a program that's through the CSUs called the CERN Bridges program. And it's all about STEM research, and it funds you for a year to work full time in a lab that's using stem cells to answer questions. And that's how I ended up at Salk in the Gauge lab. And then once that program was over, that year was up, I got my job at the core, and here I am.
Yep. For those of, I know some of you might be joining us from not in California. CSUs are the California State University system. And there is a large bit of funding coordinated through the state, through the California Institute of Regenerative Medicine, that sponsors hands-on stem cell training programs through our Cal State University system. And both Amy and I are products of those training programs. So they've been around for quite a while, and they're very awesome resource in the state. These type of programs are a lot of different places though. And so if you are interested in stem cell sciences, regenerative medicine type technologies, there are probably training programs in your local colleges that have opportunities for this. And would encourage everyone to keep an eye out for those as well if you are more interested. Also, internship opportunities for high school students and other things are present. Monica plugged the high school internship program that the Salk sponsors every year. We always host 1 or 2 students in the core for that program. And so these opportunities are present. You don't have to wait until you're in college to get some experience and insight into whether or not these are things that actually interest and excite you. It's something you might want to do more professionally. How about Jeff? What initially sparked your interest in a scientific career?
I think I've always kind of been interested in STEM-related fields in general. I would say the big moment that I made a decision was when I was in high school, actually. My mom got breast cancer, and I remember taking her to her treatments, and I asked the oncologist, what is cancer? I didn't understand what it was or how it worked. And I decided I need to learn more about biology. And then in that process, I started reaching out to professors, and eventually in college, I started volunteering in labs to understand the complexity of cells and how they operate. And it turned out to be really fun. You could be very creative in science, and you get to ask really interesting questions, kind of probing at the nature of the universe in some ways, in a very small way. And since then, I've kind of been just going with it. It's been a great, fulfilling career.
Yeah, awesome. Thomas, how about you? What initially sparked your interest?
That's a dangerous thing to get me talking about because I'm all about it. I have always really enjoyed problem solving, and I've always been really curious. And for me, science is really one of the best ways you can be curious. You have so much freedom, like Jeff and I have talked about, and even Ken and Amy experience every day, is developing tools. What tools do you think are important? What tools do you think we need? What tools do you want to develop? And for Jeff and I, it can be what diseases are we interested in? How do we want to investigate those diseases? What do we think the answer is? There is no right answer. So you get to decide what the answer is. And the more creative you can be, for example, some crackpot scientist at Harvard deciding she was going to make 3-dimensional balls of cells and turn them into brains is how we got the entire organoid technology. And that took someone to say, what if we did this, And I think it's really one of the best career paths where you have that control to express yourself and kind of choose your own fate and choose your own destiny.
Yeah.
Plus, you get to work when you want with the cells you want. Unfortunately, stem cells kind of make some choices for us, like working on the weekends occasionally. But I have unashamedly shown up to the lab in pajamas, in all sorts of mental states, tired. Sometimes we'll hang out and we'll just complain in the back of the lab. Ken, Amy, and I'll talk, have some snacks. There's always a candy bowl. It's like the best work-life atmosphere you could ask for. At the end of the day, you're all working as part of a team to solve these issues. So everybody can complain and say, man, my experiment didn't work. I'm so bummed. You'd be like, mine didn't either. Same boat. We're still here. It's late at night, or, we had a great day with some great data. It's super exciting to get closer to the answer. And it's just really invigorating and fulfilling in that way.
But at the end of the day, what really drove you there is the candy bowl. Is that what we're going to leave that? Okay. It was the candy bowl that did it. Awesome.
I got in trouble early. I can't believe
we should make sure that thing is well stocked, Thomas. Okay,
you can actually see it. It's right behind me.
Yeah. Oh, there it is. We featured it unintentionally. How about continuity there?
Celebrity.
And it is full. We're doing our job.
Yeah, I'm not making this up. You can see it on top of the microwave.
Okay, so we have one last culminating question here. And what advice would you give a high school student that is interested in a STEM career? Amy, I think we'll start with you again.
Well, there's a lot of high school programs now, like summer programs, and I would investigate what are available near you and be productive with your summer and do something. Or even if it's just volunteering for whatever you are able to get into, but just jump in and try to get into somewhere and see if that is where you want to be, like if it's for you. And there's a lot of different type of science that you can go into. You don't have to do stem cells. You can do ecology or oceanography or whatever interests you. It's all science and it's all fun.
Yeah, I 1,000% agree with that. And I'll add two things. One, kind of to the point I made earlier, where I wish I had been more proactive in reaching out to people early on. And that is what Amy's talking about. It's 100% that. Don't be afraid to reach out to a lab that you might want to volunteer in. The absolute worst thing they're going to do is say no, right? Mm-hmm. And so don't be afraid to do that. And then the other thing is, don't worry about, getting into something and then realizing you don't enjoy it, right? You have done a great thing in that, you now know that this is not what you want to do with your life, right? And that is perfectly acceptable outcome. And the worst case scenario of that whole situation is that you have spent, 2 to 3 months working with some really awesome people and have made some great friends and connections that you will have the rest of your life, right?
Yeah.
So don't be afraid to, try something and hate it. That is perfectly fine too. And I tell every single intern we have in the lab that, if they leave this and hate stem cells, that is perfectly fine. It's that they're here and getting the experience that matters, and whether or not that is impactful for their future and something what they want to develop further on is, up to them. Right. And it's okay not to.
Thomas, there's a lot of,
Amy.
Sorry. I was like, there's a lot of things that overlap in the different fields of science that if you know you don't like stem cells, you'll still gain experience that'll translate to other fields. So.
Absolutely. So many transferable skills, just how we think and kind of some of the basics of how you do science is really transferable to a lot of different arenas, in and outside of STEM even. Thomas, how about you? What's one bit of advice you would give to a high school student interested in a STEM career?
I want to echo that completely. Odds are you will not like your first experience. You have to find something you fit into.
Yep.
And I've had friends try 3, 4, 5 different labs and be like, wait, maybe I don't like science. Maybe this isn't the field I should be in. And then they get the position working with something that they're really interested in. And it changes everything. So much of it is about what interests you and what you're passionate about. You need to try multiple different things, multiple different labs, different techniques, different tools before it clicks. And it's something that you don't really know or something you can't really name until you've done it. And my friend now, she really thought about dropping out until she came across a lab that makes these really weird genetically engineered mosquitoes that can that help stop spreading malaria. And they were developing these wild genetic tools that didn't exist before. And she was just so interested in the fact that this directly could change the lives of so many people.
Yeah.
Thinking about places where malaria is a huge issue. And on the other end of the spectrum, I'm working on projects that won't help people for years, for 20, 30 years. And for her, the key was being on this project that's like, we could deploy this next year if the tests keep going well. And that changed her entire outlook and investment. So I really encourage everybody to try multiple different things, because you're going to come across stuff you don't like until you come across the thing you love.
Yeah, that's totally it. And when you find that thing that just hits that spark, you're not wondering, right? You're just like accidentally staying up to 3 o'clock every night reading stuff about it and like devoting every ounce of your energy and attention to it. And then, 15 years later, you're directing a stem cell core. I'm like, what happened? How did that happen? How did I get here? Hey, Jeff, how about you? What's one bit of advice you would give to a high school student interested in a STEM career?
Well, I totally agree with everything that's been said so far. And I think generally push yourself outside of your comfort zone and don't be afraid. Don't listen to your self-doubt. You can do it. You're smart enough that no one's asking you to be an expert overnight. The nature of the job is learning. So we learn these things slowly over time. So don't be afraid if, your math is weak right now. Work on it. You can learn. You can do this job. Go for it.
Yeah, and on that math point, if you guys were paying attention during the video, I definitely said 9 times 9 is 89. So maybe you don't need good math skills after all to be a scientist. Maybe you can get really bad at your multiplication tables and still get there. Just so you know, I do know it's 81. I want that on the record, and hopefully we can update that video at some point with some voiceover or something. Okay.
Thank you so much, all of you. Ken and Amy, thanks for giving us a sneak peek into how the stem cell core works. And Jeff and Thomas, thanks for giving us a little bit more about your research. It's been great to hear how you all work together, and really the advice that you've had for all of the high school students has been so fantastic. So thank you so much for being a part of the stem cell core panel. And Josh and Madison and I really want to thank all of you for participating in the virtual March of Dimes High School Science Week. This may be the first video you watched, or maybe you've watched all of them. So we hope you have a chance to go ahead and check out some of the other panels. If you haven't seen any yet, you may be sparked by something that is very interesting to you. So thank you so much for being a part of the week, and we're really looking forward to having you apply for one of our programs. We hope to see you at the Salk campus soon. Thanks, everyone.
Thank you.
Thank you.
Thank you.